Drug discovery is basically a mechanism for identification of drugs which are used for prevention or cure of diseases. In this module, I will just give a practical demonstration how drugs works and how they identify some cavities, and then what is the effect of using those drugs to remove those activities. I will just perform a practical demonstration which is of course shown by some graphical format, and then we will see that what are different steps. So here I have given two things, one is before binding and the other one is after binding. So if I go in some details, so if I see this figure, here we have this A basically that is available and it has a cavity here. So we have a protein which is this our green part, and then we have a drug which is our red part. So drug, and then this is our protein with this complete part. So before binding of this protein with the drug, this is the left hand part, and what happens after this binding? So what happens, this protein basically moves inside this place, and when it moves inside this place, this cavity or this A basically it also removes. So cavity A is now blocked by this protein. So this is a very simple example of drug and protein binding. In this slide we have shown 3D structure of basically a protein, and then we will see what will be the effect of this protein on some structure. So this space basically it is called drug binding site in a cavity of a protein. So cavity basically means some hole or space. So this space is available which can be used by a protein to have some binding. So as we said in last slide, we have two things, proteins and then we have drugs. So in this protein we have this specific cavity. Okay, so what happens is that, as I said in previous slide, we have a cavity. So that cavity was this place. Now what happens in this is that our drug or our medicine basically binds to that protein at this specific place. And once it is placed in this place, what will happen? This drug will bind to a protein by a lock and key mechanism. So lock and key mechanism means that it will be fixed at this specific place for some specific time until we unlock this drug. Okay, so what are different steps now? So number one is ligand and protein docking. So ligand is our medicine, and then protein docking, at which place, at what stage or what location this ligand will be docked with this protein. So step one is creation of space to fit a cavity. So in this step we are just preparing these spheres. So these different spheres which are inserted in our cavity, so that these spheres can further or in future can hold our different drugs. So moving on, if I move towards next slide. So okay, what is step 2? This is ligand protein docking. As we are in ligand protein docking mechanism, step 1 was creation of spheres to fit a cavity, and now what is the step 2? Place a ligand to match the positions of spheres. So as we said that in the previous slide, we had different small spheres which are of white color in the slide. Now they are in green color. Now what we are doing, we are placing ligands to match the position of different spheres as shown in this slide. And now if I move towards third step, which is basically, so again that is just I have elaborated with the help of computers graphics, that place a ligand to match the position of spheres. So as we have these different green things, they are our different spheres, and then we have ligand to match the position of these different spheres. So if I move toward the final slide of this approach, the third step, which is basically checking of chemical complementary. So chemical complementary means that when we have put this drug, it doesn't have any side effects to other molecules. So it captures the given cavity completely, and then it remains there, and it doesn't have any side effects for the remaining parts.